next on the program, and my talk is entitled Entry of Nanoparticles into Cells. So if I could get the presentation. Okay, as you see, I'm going to talk about entry of nanoparticles into cells, mechanisms and consequences. And most nanoparticles with drugs need to be taken into cells by endocytic mechanisms. And now it's known that we do have a number of different endocytic mechanisms in cells. Here is demonstrated that you have these various forms and that the nanoparticles may use several mechanisms. And I will come back to that they can even induce their own uptake. So dynamine-dependent or independent, clathrin-dependent or independent, and you see that illustrated here. And as I said, it's getting more and more complex, and there are now different types of clathrin-independent endocytosis, and there's an increasing degree of complexity as we learn more about the molecular details. And if you want to read more about these various forms, I have a new review article which came out this year. So I won't go in detail here, but of course you have the clathrin-dependent endocytosis, but you have like the CDC42 graph1 dependent uptake and also macropinocytosis. And of course, if you go to a polarized cell, it becomes even more complicated. Then, for instance, in the cells which have been studied so far, all caveoli are on the basolateral side, and the clathrin-independent form can be regulated differentially on the two sides. Factors which regulate uptake here may not regulate anything down here. And not only is polarity important for uptake in cells, but increasing cell density will change both the cellular lipid composition and uptake from the cell surface. We measured several hundred lipid species as a function of cell density, and it's really changing. So to the students here, if they think they can delay their experiment one day and take the cells next day, the cells are very different then. And it is a challenge to study the different endocytic pathways because there are several interactions between them. For instance, there is a co-regulation of caveoli and CDC42-dependent fluid phase. I won't go through the details here, but just say that if you change, for instance, the level of phosphocaveolin, you will also change the activity of this form of endocytosis. And also, the caveoli protein cavein has been shown by Rob Parton to regulate this CDC42-dependent pathway. So, particles may even induce their own uptake and induce intracellular changes due to the multivalent binding of cross-linking or proteins or lipids on the cell surface. And we published a few years ago now that uptake of ricin B, which is a protein which is monovalent in function, coupled to quantum dot nanoparticles, these are taken up by a macropinocytosis-like mechanism in contrast to the monovalent ricin B. So coupling this ligand to a particle will change the uptake and will also affect the cell. And cross-linking of glycolipids as well can change membrane curvature. This is Shiga-toxin-induced tubule formation. It was published by Römer and co-workers in Nature in 2007. Shiga-toxin has 5 small B subunits that can bind glycolipids. So although they don't go through the membrane, they can, in spite of that, still change the membrane. And Shiga-toxin can also signal across the membrane. As illustrated here, we studied that a few years ago now, that when we cross-link glycolipids, in this case GB3, which is the receptor for Shiga-toxin, with Shiga-toxin or with antibodies to GB3, then we induce a calcium influx into the cytosol. We induce phosphorylation of the small tyrosine Syk. There is a dissociation of annexin A1 and phospholipase A2, which will then become activated, and it can then act on the membranes and form tubules, and this will again affect the intracellular transport. And that's illustrated in the next slide, where you have annexin A1 relocalization after addition of this Shiga-B that cross-links GB3 in these cells. Here is annexin A1 in green. The lysosomal marker LAMP1 is red, and here's a control cell. And shortly after addition of Shiga-B, which, as I said, will cross-link the glycolipids, you see large changes in the cell. So clearly also, if you add a lipid-binding ligand to a particle, you will get changes that you may not be aware of. And to optimize delivery, which endocytic mechanism is involved in uptake of a given particle? One may ask, is cholesterol involved, and what does this tell us? And then one should remember that several mechanisms, actually most mechanisms known so far, are cholesterol dependent. And one should remember that when you treat the cells with cyclodextrin to extract cholesterol, or the drug genistein, these are not caveoli-specific inhibitors, something one can often see in publications. So to try to help out in the field, some years ago we made this toolbox. It was published in Nano today, and down here you have the warning with methyl beta-cyclodextrin that it depletes cholesterol, but it also affects macropinocytosis and different forms of both clathrin-independent endocytosis then and clathrin-dependent endocytosis as well. And one has to be careful so one doesn't make the membrane leaky, and in order to show that, one shouldn't also only measure leakage of proteins, but leakage of ions, which will occur far earlier than leakage of the proteins. And here is shown how cholesterol may affect clathrin-dependent endocytosis, how cholesterol depletion may affect clathrin-dependent endocytosis. If you look here, we can divide clathrin-coated areas into 3 types. Almost flat ones, more invaginated, and almost pinched off structures. So group 1, 2, and 3. And in a control cell, you will have most of group 3, that's the almost pinched off form. Whereas if you then extract cholesterol, you will see that now you get more of type 1, that's the flat one. So the clathrin-coated pits become flat when you extract cholesterol. And we also found that macropinocytosis is sensitive to cholesterol depletion. Here is a control cell which takes in horseradish peroxidase, which can give a black staining. If we stimulate macropinocytosis by adding the forbol ester TPA, then you will see that you get macropinosomes and black staining inside. But if you extract cholesterol before you give the forbol ester, the cell becomes very quiet. It can't ruffle anymore. So clearly extracting cholesterol will affect different forms of uptake into the cell. And importantly, particles may end up in a different location than the free ligand. So you can't think that you direct the particle to the place that the ligand normally goes. For instance, transferrin coupled to Q dots are endocytosed via clathrin-coated pits, but they do not recycle. This could be size-dependent. We never investigated that. And also, Shiga-B in itself can go to the Golgi and then to the ER, but if Shiga-B is coupled to Q dots, they don't go to the Golgi. I will show that in a minute. And also, particles may affect other pathways in the cell than those they use, perhaps due to trapping of membrane. So how can one make sure that a given ligand and not a nanoparticle is internalized by the cell and not only absorbed to the cell membrane? There are several possibilities. We can do EM with serial sectioning. One can do EM fixation in the presence of ruthenium red, which stains the membrane connected to the cell surface. It gives a black staining. Or one can do confocal microscopy with set stacks. And that's what I was going to show then, that when you fix in the presence of ruthenium red, it will stain the membrane. You get this black staining. And here you see something which you might have thought was an endosome, but here you see the staining is black, so there is probably a connection to the surface. So one could easily get cheated here if one doesn't do anything more than look at a section in the EM. And here is confocal microscopy with set stacks. I won't go in any detail here. But again, in this case, ruthenium red was used again. And here you see something which is a pinched off structure, an endosome. Here the membrane is not stained with the black staining, and you can see internalized gold particles. And of course, with high resolution microscopy, then one can more easily identify what's in lysosomes. For instance, here is a lysosome labeled with LAMP1, and you see some red nanoparticles inside. So due to the, I should add also something about escape of nanoparticles from endosomes. How can that happen in case one wants that to happen? They can escape by photochemical internalization, which I will mention, and it has been published that one can get escape from endosomes with positively charged substances, or one can utilize penetration properties of virus and toxins. And I will just illustrate the photochemical internalization. Here you have a target and a drug and the photosensitizer, which then is internalized in the endosomal membrane, and you get formation of an endosome with the drug. You shine light on the whole structure, and then the endosomal membrane is disrupted, and the drug can find its target. So I think that today one can say that cell biological background is important to draw the right conclusions. But what is really the case is that interdisciplinary collaborations are essential, because both the chemistry and the cell biology are getting complex, as well as other methods used. And we have a national competence building project in Norway headed by me, and I'm not sure if the poster is up. It's up, okay. So in case you want to know more about that. And here is the group, a group picture from last year, so we have to take a new one. And collaborators, briefly mentioned a group in Oslo and several international collaborators, both when it comes to nano and cell biology. So thank you for your attention. And I can take one question. Are there any?
Did you ever check the differential uptake between cells? I mean, for example, hepatocyte and copper cells, are they behaving the same way to take up the nanoparticles? Then how those nanoparticles with the same size and with the same modified surface are taken up by sometimes copper cells, in other cases hepatocytes? And do you have any?
I haven't looked at those, but I'm quite sure that you would get different results depending on the cell line you look at. Because, I mean, some cells have much more ruffling than others, and even the number of clathrin-coated pits can vary a lot between different cells. Some cells have a lot of caveoli, like endothelial cells. Others have hardly any. So, okay, I think we should continue there.